Hi. I have two questions. I had to step out for a minute, take a quick emergency call. The first one with the arm. Well, both are related to the arm. First one is I didn't see. I'm guessing it's waterproof, or there's some environmental considerations to it, and that's the first one. And the second one, it's still under investigation. What do you foresee it's going to be the affordability of this arm?
Okay, so the first question about waterproofness, almost no prostheses are waterproof. So even any commercial thing that is waterproof. But what you can do, we have tried, yes, environmental considerations are definitely there. So for example, in the box that we have put in there, we have a little lid on top, which is a silicone lid. But if you just walk out in rain and deluge it, it would not be okay. The hand that we have is a commercial hand. That commercial hand is not a waterproof hand. There's a new company called Tasca in New Zealand that's making, it's still not waterproof, water resistant. They're making that. But there are issues with the connections that you would worry about. So unfortunately, we do not have commercial-based systems that are fully waterproof. With regards to the affordability is a mixed, so there's multiple components to this, right? So there's components that is the cost of the hand itself, the orthotic and prosthetic fitting process, which happens with the prosthetic clinic, and the implant itself. This implant is built off of the cochlear implant. So if people have seen people who are deaf, who have lost hearing, and so it is built on it. The cost of that will depend very much on how many people use it, which market you are going to be in. The same cochlear company sells its devices in the US at one price, at a different price in India or in a developing country. And so that is going to be part of the issue that we have to look at. And as more technologies come through, more neurostimulators are developed, then and if the application can be to more than one thing, not just for the arm, but the same technology can be used for the leg, which is what the whole idea here is. We are stimulating nerves. We are getting some signal, taking that signal and stimulating nerves. So the whole idea is it's a platform technology that could impact other things. Then you are getting into a larger production. But otherwise, it will be an expensive effort.
Okay, good morning. I have to tell you, I would use the hat making thing in my classroom.
Great.
It's really good. I have a very specific question for you. Would you consider taking Miami-Dade college students as an interns in your group?
Absolutely.
Thank you. That is a big applause. I haven't finished.
Okay.
What about professors?
Yeah, actually, that is also, almost 60% of the students in biomedical engineering are transfer students who have completed their AA degree. We want them to come prepared. If there is something, we've actually been having this discussion that we want to make sure that whatever is the initial curriculum that we offer at FIU for the freshmen, that the transfer students should be prepared with that so they hit the ground running. They don't have the luxury when they step into engineering of saying, okay, I'm going to take this class again or whatever. So the more we can do to partner, make sure we are clear in communicating with all of you of what is needed, that would be great. We are going to have an undergraduate research day in a few weeks at the department level, and of course at the college, and college and university level, there are things. So I'm looking at these and saying, hey, these guys do a lot of research. This would be fantastic. But maybe we can explore how we can look at, while you are still a transfer student, and partner with the labs that, at least I can promise biomedical engineering, right? So in that, and because BME is life sciences and engineering and physics and chemistry, all of it, it's broad. So we should talk, yeah. Okay. Anybody else? Yes.
Good morning, Dr. Zhang. My name is Zohaib Khan, a former student at the Miami-Dade College. So I just had a question. First of all, I would just like to say you're doing a phenomenal job, very impactful job. So I would like to thank you for that.
Thank you.
So I had a question about like I know that it's not commercially available, but when it becomes commercially available, how affordable and easy do you think it is accessible to anyone?
So I answered the affordability question. The main thing we have to get to is, like most devices that are used for rehab or whatever, we have to get Medicare, insurance companies, all of them to reimburse the provider. So that is the pathway that we have to go, and we're already looking at that to say, can you, because insurance companies pay for the prosthetic hand. They don't always approve the same prosthetic hand for everybody. So we're going to have to work this system to do that. The other pathway is, the way we have designed it is that our implant system can work with multiple prosthetic hands. So if different companies were to sensorize their prosthetic hands, we have to change some electronics, and we are actually right in the process of now working on that part to make a universal adapter, so to speak, So then multiple prosthetic hand companies could work with it. So if it is of improves quality of life, and that's why you need to do these clinical trials to say, if it is improving people's lives and they all say, I want a sensorized hand because I am going to get this implant, this really changes my life, then that story changes of, does it matter that if you have sensation, are you going to do work in the workplace? Is it going to have an economic impact? Versus that you can't do half the things that you are capable of doing because you don't have a hand that can feel. So those are the arguments we are going to have to put in place for reimbursement to occur. So there's a whole another aspect of reimbursement that we need to tackle, and that is insurance companies, medical places, medical device companies. So that is the entire another picture.
Okay, we're going to take one more question from over here from Dr. LaBerge, and then Dr. Young is going to be available. I know you all want to talk to her. She's amazing. So just bear with us for time.
Yes.
Thank you for your presentation. It was excellent. I wonder about your partners, and that is, when you create your partnership in something like this, a big partner for you could be the VA. Yes. And that, or various amputee societies. Are they part of your big partnership, or is there some sort of priority for the servicemen that come back and in getting this kind of?
So the VA. So let me step back and tell you that part of the funding that came out of this was from the Department of Defense through DARPA to start the clinical trial. And you guys are going to be the first ones to know this, but I just got word, literally yesterday, that the U.S. Army is going to now fund us for the next part. And that funding is going to help us partner with the Walter Reed Military Medical Center in DC. There's lots of steps between now and that, because you need approval from the FDA, regulatory bodies, to say, we're going to go beyond here to another site. It's not just here. But that is the hope. That is the idea. So National Institutes of Health paid a lot of funding to get the technology developed. DARPA, along with US Army, helped start the clinical trial. National Institute of Health has come back to expand that enrollment of clinical trial, and now literally a day or so ago we hear from the US Army that they have accepted our grant for the next part. So yes, servicemen and women are absolutely important right up front, but also veterans and the rest of the population. We need to give solutions. So, thank you very much, and I'll be here.
Thank you again, Dr. Jung. If I may now ask our esteemed college president, Dr. Rolando Montoya, and the Padrone and North Campus's president, Dr. Malluci Harrison, to please join me on the stage as we offer a small token of appreciation for Dr. Jung. On behalf of Miami-Dade College, the School of Science, and our students. We will now begin our oral presentations. Please welcome to the stage Dr. Jorge Ober, a celebrated member of our biology faculty. Dr. Ober will serve as today's moderator. Dr. Ober.
Good morning and welcome. It is once more a pleasure for me to be facilitating the morning session for our student presentations. The best thing that happened this morning is walking in and seeing my alumni who have come from out of state to participate in this presentation. So, applausos to you. So before we start, a few housekeeping things. I like to thank once more. I know Dr. Montoya already said so, but I'd like to thank once more those people who make this possible, actually. The grant institution, the U.S. Department of Agriculture, the National Science Foundation, to the many mentors and teachers who spend hours training you to make a better person, a better professional, to the people who sit down to write the grants weeks and months in advance under high pressure. Those are the people that make the money possible, and the administrators who actually support our work. So kudos to you all. I also like to thank our partner institutions, past and present. For example, we have here the Florida Atlantic University, Nova University, St. Thomas, and also in the past we had UM as well. So these are institutions that make possible, and also FIU, who make possible for students to go out from our home base here at Miami-Dade College and do research at other laboratories. Before I continue, let me plug in a very important piece of information. During the 2 weeks preceding the Thanksgiving holidays, we're going to have here at the North Campus, during our activity hours, presentations on our own research grant sponsor programs for our students, and also the opportunities from the research for experiences for undergraduates that are sponsored at the NIH. This allows our students to go outside the state to do research. They're fully paid, anywhere from $3,000 to $7,000. They'll pay for your room and board, and anywhere from 6 weeks to 12 weeks. So it's very important that you're aware of this. Only in biology, there's over 175 institutions that are providing these opportunities for you, as many in chemistry, physics, or sciences. So be aware of those, because the application starts in January, and by April, they're usually telling you who has been accepted. By May, you're out already in the field, May and June. So make sure that you're aware of those. We're delighted to have students actually use of these opportunities. So let me proceed and give us some housekeeping rules. We have 5 presentations, so the students are going to have like 12 minutes to present. I am going to actually give you a 5-minute warning. I'm going to go like this, 5 minutes warning. If I come behind you, it means it's over. So then we're going to have microphones around the room for the audience to do questions. We're going to take around 3 minutes for questions, and then we'll move to the next presenter. After the end of these presentations, you're welcome to meet the presenters and look over their posters. We're going to have 2 sessions of 30 minutes each. Dr. Okafor is going to explain a little bit more about the process. And please stay until the end, because we have some good prizes and awards for our students. Okay, without further ado, let me present the first student presenter. Franklin, are you here? Okay, come on over, Franklin. Applause for Franklin. So Franklin Bello is working towards his AA with a pathway in computer science at the Wilson campus, and conducted his summer research under the mentorship of Dr. Quezada on the development of an algorithm for feature extraction on respiratory signals using mobile technologies. He is president of the Wilson Campus Math Club and peer academic leader under the STEM Archos program. Please welcome Franklin Bello.
Thank you. Welcome, everyone. Good morning. So, my name is Franklin Bello, and alongside Dr. David Quezada, we have been working for the past couple of months into the development of a system that will help people track their respiratory health. What I will show you today is the progress that we have so far. If you have any questions, please keep them to the end. I am more than glad to answer them. Without further ado, let's start. So, Mobile technologies play a fundamental role when it comes to medical surveillance. In fact, applications capable to track respiratory conditions, they are increasing in popularity. Respiratory conditions are often triggered by certain factors which vary depending on the situation. For example, the weather is one, the pollution, or even some food allergies. Surveillance programs that keep track of these factors are super important because okay. It's super important because what people do nowadays to keep track of their respiratory health is that they either go to see a specialist or they have to acquire expensive sensors, which are not precisely the most comfortable to wear. The ultimate goal of this project is to develop an app that fuses the weather and vital signs to determine how propense a person is to contract a respiratory disease in the near future. This will be done by performing a spectral and acoustic analysis of a respiratory signal recorded by a mobile device. Based on medical literature, there are two types of respiratory patterns: normal respiratory patterns and abnormal respiratory patterns. For the sake of simplicity, we're going to focus on the first group. And what characterizes these is the number of full breaths per minute. There is a distinction between breath and full breath. A breath can be an inhalation or exhalation, whereas a full breath is a combination of both. So if we take into account the breathing rate, then we can quantify these patterns. For example, apnea is considered as the absence of breathing. Euponia, when you have a regular breathing rate of between 12 and 20 full breaths per minute. You have bradypnea, which is decreased breathing rate, less than 12 full breaths per minute. And finally, you have tachypnea, when you have more than 20 full breaths per minute. The ultimate goal of this algorithm is to determine the breathing rate, because this way we can classify the respiratory patterns into these groups. Now, the process to develop the algorithm is the following. First, we need to record our signals. Then we pre-process it or clean it. Then we process, we extract the important features. Then we classify the signal. And finally, we implement this in a supervised learning algorithm. Four students volunteered to be part of this experiment. The four of them are students at Miami-Dade College Wolfson campus. And before we started recording the samples, we asked each student to fill a brief questionnaire about their respiratory health. When we collect more samples, this will be useful to determine and notice patterns between each and one of the students. The recordings happen in an empty classroom with minimal noise. As you can see in the right picture, the setup was as follows. The student is sat on a chair, and he, in this case, has the microphone at a distance of roughly 1.5 feet from his mouth and nostrils. We asked the student to breathe normally, first through his nose for around 1 minute, and then do it again, both through his mouth. Once that we recorded the signal, it's time to clean it. And for this purpose, we utilized this open source software, Audacity. Not only because it has a great documentation, but it has two important features that will allow us to do the job as easy as possible. These two functions are the noise reduction and the filtering. For example, here we have one of the signals that we recorded right after being imported into Audacity. As you can see in the top, in the waveform, it is hard to distinguish the breath from the noise. And if we take a look into the spectrogram in the bottom, if we look at the spectrogram in the bottom, there is this blue shaded color consistent. This is basically noise, and we want to get rid of that. How the noise reduction function works is that we leave the initial seconds of the recording into relative silent. This will capture the noise profile, and once we send that as a parameter into the function, it knows what to get rid of. So we go from this into this. We no longer have the noise, and we can distinguish clearly the readings. However, we still have some sharp peaks, and this is consequence of the residual noise in the lower frequencies. So to get rid of them, we just use a high-pass filter. Now, we no longer have the peaks. We have a clean signal, which is ready to be exported into MATLAB. As I mentioned, MATLAB was the preferred environment to perform the processing. And here, our purpose is going to be to determine the number of peaks. And afterwards, we will determine the reps per minute. First, we will have to downsample the signal. This will help to reduce the processing time. However, we will not longer be able to perform a spectral analysis. Then we get the envelope, which is a curve that describes the behavior of the signal, and then we locate the peaks. For example, here's the signal that we preprocessed right after being imported into MATLAB. It has a sampling rate of 44,000 hertz. That means 44,000 samples in 1 second. The longer it is, the more samples, hence more processing time. After we downsample it, now we have 44 samples. This is going to be better for our processor in our mobile device to work with. Then we get the envelope. The envelope, again, it just describes the higher and lower points of the signal. That's it. And MATLAB offers different ways. However, after trial and error, we determined that the best way is to use a moving window root mean square because it is efficient and it gives the best behavior. However, it needs to be smooth, and I propose 2 different ways to smooth the signal. The first one is obtaining the peaks envelope. This is done by calculating the how do you call this? Sorry. Okay, yes. This is done by getting a spline interpolation around the local maximums in a certain window length. And after we smooth it, then we can locate the peaks. And for this analysis, we determine that the system predicts that the person breathed 27 times in 1 minute. Now, this is counting both inhalations and exhalations. If we want the total number of full breaths, we have to divide this number by 2. So it will go from 27 over 2 is going to be 13.5. So if we round it, it's going to be 13 full breaths in a minute. So it will classify as an eupnea, a regular breathing rate. The second method is using a smoothing spline, in this case with a parameter of 0.9. And again, it produces a smooth curve. When we locate the peaks, it determines that the person breathed 28 times. If we round it again into the full breaths, it will be a number of 14. They produce different results. We repeat this process for the other 7 signals, and we can construct the following tables. Just to keep it simple and straight, the peaks envelope method, 5 out of the 8 tests were successful, where for the smoothing spline method, 6 of the 8 tests were successful. Both tests failed when the person breathed more than 30 times in a minute. However, for the smoothing spline, if we just increase the smoothing parameter from 0.9 to 0.99, the test is successful. So 8 out of the 8 tests will work properly. To wrap up, there are two major differences between each of the methods. The first one is the processing time. If we use the peaks envelope method, it takes an average time of 25 milliseconds to process, whereas the smoothing spline method takes 261 milliseconds. That's 10 times slower. However, the second method is more accurate. There are 2 limitations to the algorithm. The first one is the noise. The major complications that we have during the recording was that a student was screaming at the end of the hallway, or was slamming a door, or we were in downtown, so there were a lot of cars honking. So those are things to take in consideration. Not all the environments are perfect. And the next one is that this algorithm is unable to measure intensities for that same motive. Further work should be aiming to record more samples and get more numbers. And we want to implement a voice activity detection algorithm. This will do the job from importing it into Audacity and exporting it automatically. We will combine acoustic analysis with spectral analysis, and instead of using a breathing rate, we want to use images from the spectrogram to train the neural network. In conclusion, acoustic analysis is really susceptible to noises, and since we live in a world, in a city so populated like Miami, it is not conventional to do it unless you have a controlled environment. The peaks location algorithm gives a reliable number of peaks for Oh, okay, sorry. So yeah, this peaks location algorithm is considerably reliable for regular paced breathings, and we need to collect more samples in order to come to clearer and more concise conclusions. Finally, the first method has shown to be more faster. It has shown to be faster, but less accurate than the second method. Finally, I just would like to thank my mentor, Dr. David Quezada, for his unconditional support and guidance throughout the entire process. To Dr. Margolin, she helped me a lot to set up this presentation. And to the 4 volunteers who I was every day behind them, hey, can I record your breathings for my research? And finally, to the Archos program for giving me the opportunity to be here in front of you guys presenting my work. And with that being said, that was all my presentation. Thank you very much, and if you have any question, I will be more than glad to answer them.
Thank you, Franklin. Thank you so much, Franklin. Do we have any questions from Franklin? Okay. Go ahead.
Lady first, obviously.
Okay.
When I worked in the Miami Jewish Alzheimer's unit as an EKG technician, when we, like record respirations.
Excuse me, I cannot hear you clearly.
When we record respirations,
Mhm.
we usually don't tell the patient that we're recording the respiration because it changes their breathing habit. So how did you try to avoid that error when you requested that your volunteers record, that you're going to record their respirations?
No, what the setup was like is that I will start recording beforehand, and I will just put it on the table. Put it on the table, and the patient, I just ask him to breathe. Just breathe as you would normally do. I mean, we did let him know that we were recording. However, I didn't let him know which part of the recording I was using for the experiment.
Thank you. Professor, one more?
Sure, thank you. Really quick, here, hey.
Hello.
Hey. An envelope is, as you mentioned before, is a function that evolves, wrap the highest frequency signal, but that offers long-term information. I didn't hear any conclusion about the envelope analysis. What did you get out of that, the analysis of the envelope?
No, we mostly use the envelope was to locate the peaks of the function. We didn't use any frequency component from the envelope in order in the final result.
One more?
Can I add something to answer?
We have a next speaker, so I think...
I mean, if there is no question
if there's
left.
questions, you can... If there's no questions, I think we have one more.
Is this going to be open source?
Yes,
Done?
we intend this to be open source and available to everyone.
All right, thank you.
That's one of the reasons why I'm explaining step by step how the algorithm works instead of by wrapping it up.
Thank you so much.
No,
You'll
thank
have
you,
the
everyone.
opportunity to ask questions in an hour or so. So as a reminder to our speakers, when I give you the 5 minutes warning, instead of bogging down into detail, you will have the opportunity to ask the speaker questions about the process and the protocol in more detail if you are so inclined. So that's in order so we can accommodate all the speakers. Our next speaker is Doris Cruz. Doris, come on around. Doris is pursuing an AA with a pathway in biology. This past summer, she conducted research under the mentorship of Dr. Francisco Koro on bioacoustic at the Padrone campus, Miami-Dade College. Doris is passionate about continuing to participate in research and planning on earning a bachelor's degree in biomedical engineering. Please welcome Doris.
Good morning. My name is Doris Cruz, and as he said, I'm from the Eduardo Padrone campus. So today I'll be delivering a presentation which is called The Impact of Digitizing Acoustic Waves on Scientific Research, a case study. What is an acoustic wave? So an acoustic wave is a mechanical wave that consists of the sequence of pressure variations of the medium whether in which this wave is propagated, whether liquid, solid, or air. An acoustic wave can be described by frequency, intensity, and direction and temporal patterns. What are the detection and recording methods for acoustic waves? So for detection, we have the microphone for air propagation. The microphone is a transducer because it converts the acoustic waves into electrical waves. Over there, we see, I'm pointing this way, the wave, how it comes into the microphone transduces into the electrical waves, and then it's amplified and finally recorded. For recording method, I'll be talking about the classical recording method. The classical recording method is called the magnetic recording method. How it works is storing the electrical signals in magnetic fields. Over here, we have an example of an old classical recorder. You remember your MP3 players, your classical iPad, let's say. But this recording method only worked for low frequencies, like the human voices, for cricket and frog emissions, music. It didn't work for high frequencies, like bat and moth emissions. And for the addition purposes of these recordings, it was very time consuming. It would take a long time to edit, so we couldn't study them as much. Okay. So what's the difference between an analog recording and a digitized recording? So the classical method of recording, the magnetic recording, would store the signals analogically. So the wave would be a continuous wave that replicated the original acoustic wave. The new technology of digitized recordings consists of sampling the analog acoustic wave with at least double the frequency of the acoustic signal. So now instead of having the original acoustic wave, we have a set of numbers, therefore the name digitized, and it's stored in the computer file. Which this one is way easier to edit, like our last presenter said. It's easier to edit, so that's the big benefit of having the digitized recordings. This is how an analog and a digitized wave would look like. So how I implemented this in my research for the summer. So a previous background on the literature about my research, it's in 1990, Sandefur and Connor, using analog techniques and magnetic recordings, they identify and describe the acoustic emissions of a Floridian moth species, commonly known as the polka dot wasp moth. Its scientific name is the Symptomatic epilase. I'll be referring as S. epilase. They were the first ones to demonstrate the need of acoustic communications during courtship behavior in the same moth species, but with this moth species only has 2 cell ears. they also suggested certain features of these emissions, but were unable to demonstrate its biological significance due to technical limitations that they had back then in 1990s. This is how they looked at the emissions from this moth. They distinguished that there are acoustic emissions, named modulation cycles, that the passive half modulation cycle, which is this one back here. I don't know if everybody can see, I'm pointing this here. The second one is the passive, has a greater intensity than the active half modulation cycle. This is how a normal emission, a normal signal from the SPLIs would look like. So my research group, they analyzed and digitized recordings and were able to edit and alter the natural emission, the natural signal, and they placed the passive half modulation cycle in front of the active half modulation cycle. This is how it looked like after we edited. This allowed them for description of new unknown features of the acoustic emission of this moth and their courtship behavior. So in this summer, I used Bat Sound, which is a software, and along other computerized programs to analyze and process digitized acoustic waves as we can see here from the SPLI's females. We edited them, and how it looked like we substituted the active half modulation cycle with a passive. So we call this the passive modulation cycles. And we emitted this to the moth to see the response. Our aim was to see if the moth was able to identify whether this signal was modified. Our findings. So at the end of the summer, we concluded that the SPLIs respond with less variation coefficient, a higher response for modulation cycles per series, and shorter latency to the active passive. So they responded to a normal stimulus better than the one that we already had edited. So at the end of this summer, I realized that without this technology, these new improvements, not only myself, but all of us here that did the research wouldn't be able to keep going and enriching mankind knowledge by obtaining new scientific data, drawing new conclusions, and generating more questions to be answered. I would like to thank the STEM space, and all the people who supported our research, including Dr. Machado, Professor Peralta, the chairperson Alfredo Leon, my team members, Jasmeli Reina, and last but not least, my mentor, which I'm really grateful for. Thank you for giving me all this opportunity, and that's it. Any questions?
Thank you so much, and we have some extra time. She was right on spot. So do you have any questions on her research? Any questions from the professors? Dr. Montoya.
Where did you get the sample of
My mentor, actually, he has them in his backyard.
What are the applications of what you have done?
Yes. So with this, we can study the behavior of this animal, and we can study its courtship behavior. Mainly, that helps. I guess in every field we have kind of something that will happen in the future. Like I guess with hearing aids, it's the same process of editing, mainly that.
Okay. Any more questions?
Yes.
used the passive-passive. Did anyone try active-active?
That might be one of the questions we're generating for next.
Okay, try one, try the other. Why not? Thank you.
Thank you.
Anyone else?
Yes, are these insects nocturnal insects, and would you expect your results to change if you measured the frequency during the day versus during the night?
Okay, so we measured these. We measured their emissions during their mating behavior, which is from 3 a.m. to 6 a.m. That's the only hours where they propagate sound. So on the daylight, they will not propagate the sound. Any more questions?
Thank you so much.
Thank you.
For those of you who would like further information on the poster presentations, Ms. Cruz posters is in page 27, and the next presentation by Rafael Mora is in page 57. Rafael, please come forth. So Rafael Mora is pursuing his Bachelor's of Science in Biopharmaceutical Sciences and conducts his summer research under the mentorship of Dr. Selwyn Williams at North Campus. He is currently a learning assistant and wants to pursue a career in the medical field. Welcome.
So good morning, everyone. My name is Rafael Mora, and today I'm going to be presenting my summer research project, which was a genotoxicity assessment of Wagner Creek Sablewood Canal in Miami, Florida. So climate change is a big issue all over the world. Recently, just yesterday, you had protests all over the world relating to this issue, but especially in Miami, since we are right at sea level, and we are already facing challenges from climate change, such as flooding, algae blooms, saltwater intrusion. So we had a lot of these issues that are affecting us. Just a couple weeks ago, actually, a grand jury reported, as you can see over here in that article, a grand jury reported that Biscayne Bay is actually at a tipping point where if any more pollution goes into the water, the damage is going to be irreversible. So we decided, due to like all this information and all this data with climate change, to actually apply this in our own backyard in Wagner Creek Sablewood Canal, which is about 20 minutes away from here. Wagner Creek Sablewood Canal has a history of being the most polluted waterway in all of Florida. So in 2003, tests were performed on the waterway that found high levels of dioxins in the water, and this was believed to be due to an incinerator that shut down 30 years ago. And you actually, in the tests that they conducted, in the lower levels of the sediment, that's where they found the highest levels of dioxins. And then in 2015, a reporter by the name of Jenny Salovich reported that more tests had been done, and they found high levels of fecal matter, heavy metals, and hydrocarbons in the water. So in July 2017, the city of Miami actually approved the restoration project on the waterway. So it took them about a year. They finished June 29th, 2018, and they dredged the whole area and they took out the sediment, right? So the goal of our research was to analyze the water and see if the level of mutagenicity in the water had gone down. So this is actually the sign on the outside by one of the testing sites that shows the name of the project, the people involved, and how much it cost. And it cost about $18.4 million. So these are the testing sites. You see Spring Garden Point Park, and then you see 11th Street up here. So 11th Street is just upstream, and Spring Garden Point Park runs right into the Miami River. So the testing sites. So this is Spring Garden Point Park, which is now called Sablewood Canal. It was artificially widened and deepened, and it flows into the Miami River. Compared to Wagner Creek, which is a lot more shallow, a lot more narrower, and it's easily accessible to the public. There's a street right there that cars and people can just walk by, which it might lead them to throw trash right into the waterway. So our methods of performing this research was we collected water samples throughout the whole month of June, and then we performed the Ames test. And for the Ames test, we used two different strains of Salmonella typhimurium, TA98 and TA100. The reason we used the two different strains of Salmonella was because one strain, TA98, tests for frame shift mutations, and TA100 tests for point shift mutations. So these are examples of the mutations. So you can see for a point mutation, where it says substitution, you have a sentence, the cat ate the rat. For a point mutation, they changed the C to an H, so now the sentence reads differently. For a frameshift mutation, you have a deletion or insertion in the actual DNA that causes the whole sentence to be misread. So you can see it either shortens or lengthens. So the actual Ames test, the way it's performed, and it's a really widely used test all across the various industries, especially the pharmacy industry, where you can test for mutagenicity. And mutagenicity is basically the ability of a substance to cause a mutation in your DNA. Now, the level of mutagenicity have a high correlation with being carcinogens, meaning they can cause cancers. They can cause cancers in our bodies. So this was the actual way we performed the test. So we collected the water samples, and then we used the 2 strains of bacteria, but the bacteria are His minus, meaning that bacteria can synthesize their own nutrients. So since they can synthesize their own nutrients, we take a strain of DNA of bacteria that it can't. It's His minus, so it can't synthesize that histidine. So in order to grow, we have to provide it that histidine. But we only provide a limited amount of histidine. So when we perform the Ames test and we put that bacteria into the water, if the water is mutagenic, it will reverse it back to histidine plus. So now it can synthesize its own histidine. And this is shown here, where you have your sterility check, your background, your strong mutagen, and your positive control, right? So if the test comes with a purple dye, so if it stays purple, it means it didn't revert back. It stayed the same His minus. But if it goes to yellow, it means the water is highly mutagenic and it reverted from His minus to His plus bacteria. So these are our results. So for the TA98 count, you can see our negative control and our actual revertant count. In our negative control, the reason we have revertants is due to spontaneous mutations that happen naturally, and they occur frequently in nature. And then you have the revertant counts. And then you can see the revertant counts are actually quite high for 3 of the dates. You have 96, 96, 76. And one of the dates, you only have 2, and I believe that is due to probably an error in actual printing the sample. And we can compare our revertant count to our positive control. And our positive control basically comes from we use a substance that is known to cause mutation in the bacteria. So those should be the highest levels of mutagenicity. And you can see for 2 of those dates for the TA98, it matched our positive control. So then for TA100, same thing. We had our negative control that had a baseline of about 24, but our actual reverting counts were 96. So they all contain high levels of mutagenicity, meaning this water is heavily polluted. So these are the same results I showed before, just in more visual format. And you can see consistently in the red and the orange how high the levels are of mutagenicity. And then now this, so what I explained to you before were the samples that I tested, but this graph shows the samples that everyone in my lab group tested for TA98. And you can see the positive control in purple down here in the bottom. And our actual reverting counts are in red for Wagner Creek and in blue for Spring Garden Point Park. You can see that Wagner Creek throughout the whole month of June was consistently high. There's a lot of pollution in Wagner Creek compared to Spring Garden Point Park where the levels kind of fluctuate, right? And then for TA100, same thing. TA100, it was consistently high, but Spring Garden Point Park, you do have those fluctuations. Now, the reason for these fluctuations we believe to be due is that there's differences in the 2 sites. Spring Garden Point Park is a lot deeper, it's a lot wider, it dumps into the Miami River, and the pollution can be diluted. Compared to 11th Street, where in 11th Street, it's a lot more narrow, it's a lot shallower, so the pollution can actually be highly concentrated. Also, in 11th Street, there are a lot of businesses and industrial things and hospitals in the back. So and a lot of streets, a lot of roads, a lot of highways. So all that runoff can potentially go into the creek and cause a lot more pollution in the actual water. So for my conclusion, we believe that either the restoration wasn't as effective as we previously thought, or there are still a lot of sources of pollution that are getting into the actual waterway. So the next steps is that further research needs to be performed in order to assess what are the sources of pollution, but also what are the actual chemicals in the water causing the mutations in the bacteria. So I'd like to thank my mentor, Dr. Selwyn Williams, my lab group, Ariana Johnson, Herlane de la France, Seema Ganavati, Malika Ganavati, and Luke Raymond, and I'd also thank the STEM Mile and STEM Engine grants. Thank you.
Thank you. right on time. We have like 3 minutes still for him to talk, so we might have time for more questions. So do you have any questions on the floor? I think, Professor?
So I thought your research project was great. I teach microbiology and genetics, and I'm like, great. One thing that I thought that maybe you're thinking about for future, the next time you do it, your samples like topped out, like they were just as strong as your controls, your positive controls. So have you thought about, so you almost can't even measure it because it's so high that it just is off the charts. So have you thought of a way for future studies to figure out what to do with your samples so that you can actually kind of get closer to an idea of what the numbers are, because they're just off the chart. They're out of range, basically.
So currently, we haven't really thought about those next steps yet, but we will probably be thinking about what you're saying in terms of maybe actually really getting a good measurement of how high the pollution is, because you're right, they are maxing out. So we haven't thought about that yet, but we will.
I'll give you a tip. Dilute the samples. Dilute them and dilute them more, and then it might just be that a lot of the water that's outside is contaminated, so you might have to have additional controls, like maybe tap water, spring water, so you start getting an idea of just how the natural samples relate when you have them in the lab, you know, with the numbers that are in range for your lab. Okay?
Thank you.
Thank you so much.
Have you shared your results with the appropriate environmental officers of the city of Miami so that they see what happened with the $18 million that they used to clean those waters?
Yeah, so we haven't shared the results yet. We're actually planning on going to the same reporter that wrote an article. But yeah, we haven't made the, because my mentor, Dr. Williams, he's been doing the research for a couple semesters. So he's decided he might go to the reporter to see what's going on.
So just in response to putting those two comments together, though, the first comment actually said that your test isn't sensitive enough to just say just how polluted it is. But you also don't present any pre-restoration data. So you don't know that the restoration didn't do anything, correct?
But
you just have very high pollution levels.
Yeah.
Do you have pre-restoration data?
No, we don't, in the same sense, we don't have pre-restoration.
So we don't know if the 18 million was a waste. We just don't know if we've moved down at all.
Yeah.
We know it's still very high.
Yeah, exactly.
Correct.
Anyone else? Anyone else? Thank you so much, Rafael. Technical difficulties here. For those of you who want further information on Rafael's work, his abstracts is in page 57, and he'll be available for questions. Our next presenter is Valeria Nasir. Valeria. Valeria's abstract is in page 24. And again, she'll be available for questions after her presentation. So Valeria conducted her summer research under the mentorship of Dr. Alexis Tapanes Castillo at St. Thomas University, and is pursuing her Bachelor's of Science in Biopharmaceutical Sciences here at Miami-Dade College. She's the secretary of the Robotics Club, a STEM IELTS Scholar, a STEM Ambassador, and a member of the Phi Theta Kappa and Tribeta Honor Society. Please help me welcome Valeria.
Thank you, Dr. Besser. Sorry. Greetings and humble salutations. Right. So I conducted my research at St. Thomas University with Dr. Tapanes, and it was on the sinus the sinus telebintifolius extracts on exhibit anti-cancer activities. Thanks. All right, so what is cancer? Before I define cancer, we've all taken basic biology, so I'd like to do a little review on biology. And no, I'm not about to quiz anybody. So let's go over mitosis. What is mitosis? Mitosis is a very necessary steps of growth and development where cells in the body divide, which leads to cancer, on the other hand, is when the cells continuously divide and divide and divide and divide out of control and forms these lumps of cells, which we call the tumors, which block the proper nutrients from going to the regular cells, and they therefore killing neighboring healthy cells. And statistics of cancer, by a show of hand, who knows anybody who's been affected by cancer? Or a lot of hands, a lot of hands. So cancer is a very, very bad disease, and it's very popular. So according to the National Cancer Institute, 439 out of 100,000 women and men get affected by cancer per year. Metastasis, what is that? Metastasis is a property of cancer, which is the very dangerous aspect of it. That's when the cancer cells spread throughout the body, which establish different little colonies everywhere in the body. Then that's what is the leading cause of cancer cells' death. All right, here we can look at the steps. The first one is just a regular, normal epithelium. The second one, you could see the mass of irregular cells. You could see the tumor cells. And the next one, they use the blood vessels to escape and spread throughout the body. And some of them exit at different sites in the body where they establish little colonies where the cancer cells continue to grow at different spots in the body. So we found this plant called the Brazilian pepper tree. The scientific name is the shiny trees, that have been, according to previous research, they have been proven to have anti-cancer properties. They have bioactive molecules that can prevent the cancer cells growth, and that's why we chose this plant to do our research. Here, the overview of the research. The goal of the research was basically to evaluate the effect of the shiny trees extract on the cancer cells' growth and migration. And the first part of the research, we had 2 different breast cancer cells line. One was called the MTF7 cells, and one was called the BT549 cells. So the first part, we basically did different experiments to see which one would be more aggressive and that are highly metastatic for this research, so we could get more effective results. So to do so, we used a technique called the scratch-based migration assay. And to do that, we grew the cancer cells on a plate with different wells. So once all the cells are in the bottom of the well, we used a pipette to do a scratch. So that creates a gap between the cells right in the middle of the well. And with an inverted microscope, we took a picture of that line. And then we incubated and let it stay overnight. And after 24 hours, we came back and took a picture of that exact spot again. And then we used different software to measure these lines to see how fast it migrates back and to close the line. And the one that was more aggressive and faster to migrate back into the line, we chose that line, which was the BT549. We chose that line to do the study with the shiny tree extracts to see how it could affect the migration, if it would slow it down, and how much it would slow it down. And we also conducted an MTT assay, which is a cytotoxicity assay, which basically tests if the plant extract would kill the cell and how toxic it is to the cells. And we also did a phalloidin staining. Phalloidin is basically Phalloidin is a dye that binds to F-actin, which are present in the cytoskeleton of our cells. So we use that technique to stain them and see how the plant would affect the migration and how the cytoskeleton would be affected by the plant. Here we could see that's the result of the first part of the experiment. So the top one is the MCF7 breast cancer cells line, and the bottom one would be the BT549 breast cancer cells line. On the left, that time 0 hours, that's right after we did the scratch at time 0. And the right side is after 24 hours when we check them again to see how fast the cells migrated back to close the line. The first one on top right, we could see that the cells did migrate back, but not completely close that gap. And in the bottom, the line is completely invisible. So that meant that the BT549 cells are more aggressive and they migrate much, much faster to close the line. And on the right, these are 2 box plots of the results that we got from the left. So the MCF7, they migrated back with about a speed of 7 to 8 micrometers per hour, as of BT549 had a migration velocity of about 27. So imagine 5 miles per hour versus 27 miles per hour. There's a huge difference. So we chose the BT549 to do the pilot study with the plant extract. So on top, the picture on top, you could see that's the one that we did with the control. So we didn't treat those cells. So after 24 hours, they just completely migrated back into the line. There's no line to be visible. And in the bottom, gratefully, we found that the sinus tree extract actually stops the cells from migrating back into the line, which is great. That was a great day in the lab. Great discovery here. And on the right, we could see the plots. With the extract, migration velocity was very, very much lower than where it was not treated. Next, we used different solvents to just compare and see how they would affect on the cancer cells migration. On the left, you see 3 box plots. That's when we use the extracts. And on the right, 3 box plots where they're just controlled with alcohol and water, where the migration velocities were super high. Next, this is the Mtt Cytotoxicity Assay. So if the plant extracts can affect the migration velocity, if it could stop them, we need to test, does it actually kill the cells, or does it just immobilize the cells and they're not able to move? So we did this assay to test how toxic it is to the plants, and thankfully we found that it's not very toxic to the cells. So these are the results from the MTT assay. So all the graphs here, there's not very significant difference between all the bars. It's between 0.7 to 1. So these are all the cells on the left, untreated, the control, and the bars at different concentration, and it doesn't really have a big difference. So that's why the extracts are not very toxic to the cells. And here, that's how we pictured and visualized the cytoskeleton on the left. The cells were not treated, and the filaments in the cytoskeletons are kind of going crazy and everywhere. That's what makes the cell, that's what proves that the cells are very mobile and they are moving and they are very spread out. And on the right, you can see the cells it's more calm, it's less filaments. So that shows that the bar extract greatly impacted their migration velocities. And in summary, we did find that the BT549 are much more aggressive than the MCF7 cancer cells line, and the experiments with the plants proved that the plants do greatly affect the migration velocity and slowed it down greatly. And they are not toxic to the plants. They are not toxic to the cancer cells, which is great. And future directions, we will do more experiments we'll do more experiments to actually identify the molecules inside the plants that actually slow down the growth of the cancer cells. And acknowledgement, I would like to thank the STEM Space program here at MDC North. I would like to thank Dr. Tapanis for taking me under her wings. And I'd like to thank the School of Science staff. I can't say names because there are so many names. But thank you. You know who you are. Thank you, everyone.
Thank you so much.
Questions?
Right on time, you have an extra minute. So this presentation brought it really close to my heart, because if I'm here today, it was because in the mid to late 70s, there was an article published in the Scientific American by Judah Folkman from Harvard University. And I was ready to do a presentation, a totally unrelated topic, and it was on angiogenesis tumor metastasis. So one of my graduate professors, I was one of the graduate taking graduate classes, and she said, Jorge, I want you to present on this topic. And I said, darn it, because I had my whole presentation already prepared. That was before the internet. That was when people used to print the slides and put it in a carousel. So it was a prehistoric era. So I had everything that had taken me weeks to do. I sat down, read that article, and I was hooked. And little did I know that as an undergraduate in my early 20s, I was going to end doing research for over 35 years of my life in metastases. And that's what my doctoral dissertation was on. So I'm thrilled that you did this presentation today. So you have a great future ahead.
Thank you.
So, yes, you never know how God works. You know, sometimes you say, oh, this is not going to work, and it's there for a reason. So I congratulate you. Do we have any questions on the floor for Valeria? Any questions for Valeria? Right there? Okay, let me go to you. And your name? Anastasia.
Hello. I have a question. Did you check how this extract affects non-cancerous cells?
Non-cancerous cells? No. This study mainly focused on the cancer cells.
Okay, thank you.
Any other question? On the back, I heard on the back.
Hey, Valerie, thank you so much
Hi.
for your presentation. I have a one question. Where do you actually see these plants? Is it here, or is it native here, or is it an exotic plant? Did you get the plant here?
Oh, it originated in South America. So from Brazil, originated in Brazil, Argentina. That's where you find the plant. That's where it's mostly abundant, but at St. Thomas University, we actually have the plant there.
Okay, the plant is grown there at St. Thomas.
Yes.
Okay.
You can see them as it's an invasive plant in South Florida. So now that we're going into the fall and into the winter, you might see some red berries around here, and they are quite invasive. I think we have two more questions in the floor.
So, sure. Dr. Nia Madison from Homestead Campus. I was wondering if this plant bark could also prevent the cancer cells from having the loss of function of cell-to-cell contact inhibition, which is also a problem in cancer, not only metastasis, but also the cells lose their contact inhibition function, which causes them to form on top of each other and form tumors.
So the question was, would that also impact that?
Would the bark extract also impact the loss of function of contact inhibition?
I'm not sure, but it should, because when we did the fluoridine staining, which the research will be ongoing, and they will do more fluoridine staining, because when we did the fluoridine staining, when they were not treated, everything was on top of each other, and everything was going crazy, and it was just spread out everywhere. And after the treatments, there is more space, and they're more spread out and not all together. So I think it could affect that also.
I believe I have one more question on the floor. Is okay? Okay, one more question.
Thank you. I'm Dr. Moll from St. Thomas University. And more than anything, I want to make a comment on the same spirit that Dr. Jung was talking about this morning. Brazilian pepper is an invasive plant. So everybody's trying to get rid of it because it grows everywhere. And it messes up our natural environments and so forth. So here's a student and a group that decided to look at the exotic plant with a new angle. What can it do as a medicinal plant? So we have to, as scientists and as students in science, look at things with a new eye, right? New eyes, and try to find alternative ways of looking at what our problems are. Thank you.
Along those lines, I wonder whether you have any information from the popular folklore or pharmacopoeia about this plant, if there has been anything done regarding probably characterizing some of the compounds, or what is its use in popular folklore?
Actually, this plant is not just anti-cancer properties that they have. They are used for various diseases. If you look it up, there are a lot of research on this plant and how it affects different diseases all over, especially in South America. Yes.
Thank you so much, Valeria.
Thank you.
Okay, one more presenter. Our next student presenter, Ernesto Suarez. Ernesto, could you please come forth? If you are interested in Ernesto's poster presentation, it's in page 10. So Ernesto Suarez is currently pursuing an NA with a pathway in mathematics. He and his colleagues conducted the research under the mentorship of Dr. Eduardo Araujo Pradere at the Homestead campus. Ernesto is a member of the astronomy and math clubs. Welcome, Ernesto.
Thank you. Thank you for coming to hear about the research that me and my colleagues did under the mentorship of Dr. Eduardo Araujo Pradere. Our team members are Lacey Abare, my colleagues are sitting over there, Lacey Abare, Denisa de Ama, and Jose C. Lopez, who was a volunteer and contributed a lot to our research, as well as Santiago Mejia, who wrote some really important code that helped us in extracting the data that we used. Our research was description of the behavior of minima and maxima FOF2 data values and comparison against IRI predictions. So we start out by giving a little introduction to what the ionospheric system is. Like the last presenter did, a little biology review. This might be a little bit of a chemistry/physics review. So the ionosphere is one of the layers of our atmosphere, actually starting at around the tail end of the mesosphere and stretching into the exosphere. It's actually the only layer of our atmosphere that has a charge. It gets that charge by way of solar radiation penetrating into our atmosphere and separating molecules in that layer from their electrons, leaving us with ions, as denoted by the name ionosphere. Now, the layer that we're most concerned with is the F2 layer, because the F2 layer contains the peak concentration of electrons in the ionosphere, which we denote by NMF2. So NMF2 we know as the peak concentration of the F2 layer. We calculate the NMF2 by the FOF2, which is the critical frequency of the F2 layer. We get the data from the stations that we analyze, but the way they get that data is by shooting up radio waves into the atmosphere at increasing frequencies, and the last frequency, and the ionosphere actually reflects those radio waves back down. But the last radio wave to be reflected back down is what we call our critical frequency, because the next one right after that would shoot right through the ionosphere and not be reflected back. And this is the equation that we use to calculate peak concentration from the critical frequency. So our research is motivated by, obviously, our mentor has done much work on studying the ionosphere He's been a professor. He's been a research scientist at Boulder in Colorado. He's been a professor for most of his life, and these are many of the papers that he's written, along with his colleagues. So the main motivation for this research is to establish a baseline for the ionospheric behavior from the solar cycles that we were looking at specifically. We want to do that because we want to compare those values to the ionospheric behavior that we will be experiencing now, since we are actually going to experience the descendant phase in solar activity. And also, we wanted to validate that the international reference ionosphere models were accurate to output for these conditions. So we use a lot of data sources, including the solar activity, geomagnetic activity, and the ionospheric behavior. The FOF2 data for the descendant phase of solar cycle 20, which lasted from 1970 to 1975, and the ascendant phase of solar cycle 21, which lasted from 1976 to 1981. Now, this is actually a new approach. We went ahead and we got the data for each day, and we extracted the maximum and minimum for each of those FOF2 data values. And that's actually a new approach that has been performed first by our team. So we also got the solar activity and geomagnetic indices for the period, the sunspot number, and the solar flux in the 10.7 centimeter region. These are the stations we analyzed. Up here we have Argentina Island, Boulder in Colorado, Moscow in Russia, and Canberra in Australia. The processing for this data was the guideline for establishing this data was outlined in this article, which was also written by our mentor. So here's a map of the stations that we analyzed. Up here we have the geomagnetic equator, which is actually different from our regular equator, which our geomagnetic equator has as you can see, a dip here, and it rises back up here. Now, the stations we analyze are specifically in what we call the magnetic mid-latitude region. The reason we use stations in these regions is because they have a lot less variability in their ionospheric behavior for when we calculate at those stations. So we decided to get two stations from the north magnetic mid-latitude region, Boulder and Moscow, and two from the south magnetic mid-latitude region, including Argentina and Canberra. So these are graphs showing the solar cycle trends for specifically our periods are boxed in red here. As we can see, this is the descendant phase. As we can see, solar activity goes down, and during the ascendant phase, which started around 1976, increased. So, for analysis, we plotted the solar and ionospheric data. We plotted the solar and ionospheric data. We calculated the trends, and we had the IRI output included. Now, the IRI is actually stands for the International Reference Ionosphere, which is the international model that is used by many companies, and is actually very important for, say, our spacecrafts whenever they do spacewalks. It's very important that they know this data, because if there's any like solar flare any high solar activity during that time, if they're doing a spacewalk at that time, they could die. Well, they probably will die. So this is the solar and magnetic activity for the descendant phase. This shows up in all our graphs for each of our stations, but since it remains the same for the descendant phase, we just showed it here to show it one time. Now, an interesting finding during this, this is the geomagnetic activity measured. This is the solar activity measured in F10.7, And here is the variability of the sunspot activity. Now, one of the interesting findings for the descendant phase is actually that, as you can see there, the geomagnetic activity saw a rise during the descendant phase. And that was an interesting finding because normally we see that value go down during the descendant phase. So here's our data for Argentina Island for the descendant phase. Here we have our FOF2, which, as I said, was the critical frequency of the F2 layer. And as we can see here, the black line here, I don't know if you can see it that well there. We have the black line there that kind of goes in a little wavy pattern, is what we call our fit for our data points, which our data points are those dots. This dashed line here, not that straight line there, but this dashed line here, it's kind of hard to see, that also goes in a wavy pattern, is the model that we got from the IRI. So this is for the maximum values, and down here is for the minimum values. The maximum, as we can see for the maximum values, one of the interesting findings that was actually one of our contributions to the IRI was that the model was not very good at predicting these peaks in the FOF2. As we can see, there's a lot of variability there between the model and the FOF2. We can see, for example, the model will peak here, but the fit will peak even higher, as well as the data values themselves. They'll peak even higher than the actual model will predict. As we can see with the minimum down there, it was actually pretty well predicted. This was for Boulder Station. Here we see the same variability between our models and our fits, as well as our data points. But it's good to note that these differences are not as big as, for example, with Argentina Island. And for this is the FOF2 standard deviations, which is basically the variability in the FOF2. This was for Moscow. See, we see the same trends here. The variability between the models, the fit, and the data values are again not as large as with Argentina Island. And we see the same trend with the minimums. The minimums are pretty accurately predicted by the model. This is for Canberra. Again, we also see the same variability as we saw with every station, which again was one of our key findings in our research. Again, with the minimum values staying relatively consistent with the model. And another one of the findings was, up here we have the maximum deviation, and down here we have the minimum deviation. It might be hard to read there, but as we see that the maximum deviation would get larger, we would see that the minimum deviation would get smaller. So this was solar magnetic activity for the ascendant phase. Here we saw geomagnetic activity go up for that period. We also saw sunspot activity go up for that period, and we saw the variability go up as you go along the time. This was Argentina Island for the ascendant phase. Here we see the same variability that we saw for the descendant phase in the maximums, but not for the minimums. Here's for Boulder. We saw variabilities, but again, as with the descendant phase, not as large as with Argentina Island. This was for Moscow. Again, as is consistent with all these plots, we saw these differences between the model, the fit, and the data. And again for Canberra. So between the conclusions, we know there's a lot of information that we had to extract from this data, including the latitudinal dependencies. One of the reasons for that stark difference was because Argentina Island was so close to the poles, was so close to the South Pole, they get a lot more spikes in solar activity, which makes it harder for the models to predict. As well, obviously, that leaves us with data quality issues that we need to identify and quantify them. we saw an interesting upward trend of the geomagnetic activity during the descendant phase of the solar cycle. We saw a lot of variability between around the fit for the max values of our FOF2 data values. And so in conclusion, we found that the IRI performed really well in following the minimum values, but very inconsistent for the maximum values. So our future research task, this research was actually presented by our advisor at the IRI task force meeting in Cyprus, in Nicosia, Cyprus, from September 8th to September 14th. He presented that research there, and they set up future research tasks, including getting data from all the stations on the map that we process in the same way. Get the IRI output for each will be included in each case. We got to classify the dependencies. We also need to do the analyses for the two latest minima that we need to complete, including the minima values that we'll be experiencing now. Once we get all the stations completed, the data quality assured, and the statistical analysis will be conducted to describe the current solar minimum. For acknowledgements, we really wanted to thank the MDC School of Science for giving me and my colleagues the opportunity to do this research, as well as the U.S. Department of Education, the STEM Space Grant, for giving us the funds to be able to do this research, and of course our advisor, Dr. Eduardo Araujo, for being an excellent advisor in helping us get more information regarding our research, and for the opportunity to participate in such a fascinating opportunity.
Thank you so much. Any questions for Ernesto? Any questions for Ernesto? We have 2 questions on the floor.
Thank you. You mentioned that these inconsistencies in measurement might have some implications for astronaut safety and space flight travel. Do they also have implications for commercial travel safety?
For commercial travel, I forgot to mention that the ionosphere is also useful in protecting us from CMEs, which are coronal mass ejections from the sun, which if we get a large enough one of those, we've actually seen those happen for example, in parts of Canada and parts of the northeast US, where we have huge blackouts because of these mass ejections, and the ionospheric behavior helps us in identifying those risks.
Okay, thank you. One more question?
We have another person, so yes. Go ahead.
Yes, to perform the statistical analysis, what software did you use?
The software, we actually got the code written by as I mentioned, Santiago Mejia. Yeah, and the program he used actually gave us those charts that I showed on there, to give us those standard deviations, as well as the trends for the solar activity, for the geomagnetic activity. So those are what we counted as the statistical analysis. So the IRI actually has a database of those data values. You just choose what days you want, what day you want, what year, and they give you all the data. They give you all the values for each hour of the day, and we got the minimum and the maximum for each of those days. Mm-hmm.
So a professor would like to add something to that.
Thank you. That was a great answer. We didn't use any pre-built statistical package. I wrote the code, and one of the students put together a Python, very simple, very clean code to run all the statistics and the plotting. The thing is, we didn't want to use any commercial because first, we don't want to pay. I don't have enough to pay for them. Second, we want to run all the stations in the map, all of them. We need a very lean, clean code to do that.
Thank you, Professor. Any more questions? Yeah. I guess we have one more question.
I noticed that most of the stations were basically that's 60 degrees north or south.
Yeah.
But you had one station, Townsend, that was about near equatorial. How would an equatorial station differ from these?
It would have more variability between our data. So
Right.
they're not as consistent as with the magnetic mid-latitude regions.
Okay, and so you said that ultimately you want to combine all stations, the data
Yes.
from all stations. So will you have outliers that you'll just not utilize? Because if it's more variable there, and also you're having issues at the poles, right?
Yeah.
So really, you really only want the mid-latitude stations, right?
Yeah. Since we saw that variability between the Argentina island was the biggest one, and we saw it was closest to the pole. That was our contribution to the IRI, so that they can improve, to help them improve their data quality.
Okay, and is there any plans on making monitoring stations? I know these are old, I get that. But is there more recent stations that we've been monitoring so that we can get a better picture of this?
That we specifically have been monitoring? No. I know my mentor mentioned to me when he was in Cyprus that he met with a lot of people from different parts of the world, and he actually met somebody from Thailand who is on one of those ionospheric stations, and that's obviously in that geomagnetic equator location. But for our research purposes, we're using the mid-latitude regions.
Okay. All right. Thanks.
Thank you so much. We are running out of time. Please, a big round of applause for our presenters. Again, they are available for questions. So we have 2 rounds of presentations, and please welcome Dr. Okafor again.
Thank you so much. Let's give them another round of applause, if you please. Before we move on to the poster session, I want to take a moment to thank all of the individuals that made today's event possible. First, the School of Science team members. I don't want to begin to mention names, but you know yourselves, you have done tremendously, and we're very grateful. We want to thank especially Dr. Montoya for gracing the occasion. He raised the level of everything. He may not know the details of all that, but he raised the bar for us, and we're really inspired that he's here. I want to thank Dr. John Wensvin as well, the Vice Provost of Academic Schools. I want to thank Dr. Harrison, the campus president for North Campus, and Eduardo J. Padrone campus. I'm going to extend my appreciation as well to the media and campus services for their tireless efforts to ensure that we have a beautiful venue and that everything runs smoothly. Thank you to Octavia Anderson and the Quick Copy staff for their tireless work on our abstract book. Thank you to the North Campus cabinet members and the student life for supporting us. And of course, our amazing faculty, for without them, the research would not be possible. I think it's a special thing to have faculty to volunteer, to partake in the kind of research that we're getting the results today. Just a little intro for the poster session. As we move on to the poster session, students, please take a look at the group letter on your name badges. Students that are in Group A will be presenting their posters for the next 30 minutes to members of the audience. Meanwhile, students in Group B will be selecting their choices for the peer award using the orange forms. At that time, Group B students will go to their posters to present, while the students in Group A cast their votes for the peer award using the yellow forms. Please deposit your votes in the boxes marked peer judging score box located in the back of the room. We're going to go to the poster session, and after that, then we have the awards for winners of posters in different categories and the oral presentation as well. Thank you.